Hello, my dear friends. It's Naik's Biology and myself Hemant Naik, your biology teacher. Today we are going to discuss about the Hershey and Chase experiment. Actually, what is this Hershey and Chase experiment? It is somewhat confusion in the NCERT textbook because it is not clearly mentioned in the NCERT textbook. But I will try my level best to make you very simple of this concept. If you are able to understand the concept after seeing this video, then don't forget to subscribe my channel. Let's start. When we will start, we have to know about the bacteriophage. What is this bacteriophage? This bacteriophage means it is these two are bacteriophage, this one and this one. This bacteriophage is nothing but a virus which is infect the bacteria. That means virus can kill, as you know very well, and they are also kill the bacteria. That's why its name is bacteriophage. Point one clear. Bacteriophage means it infect the bacteria. Now, radioactive level phosphorus, we have to try to understand. Sulfur is, if you know, sulfur is atomic mass is 32. And if you see that, if the number of the neutrons, because you know electron, proton and neutron is there, this number of the neutron, if you will change, the number of the atomic mass also changes. Now, what you did 35S, which is a very active radioactive sulfur, because the atomic mass has become changed. Here you see, if you know that atomic mass of the phosphorus is 30, but what you taken? We taken 32 atomic mass phosphorus, which having the 32 atomic mass. How it came 32? Because number of the neutrons has changed and it become radioactive. Now what happened? They make the two culture plates. One culture plate, what the Hershey and Chase taken the radioactive sulfur, one they taken the radioactive phosphorus. After that, they allow this to, what is this to? Bacteriophage. Allow this bacteriophage to mix with the radioactive elements. In this one bacteriophage, when it is leveled, means reacted with the radioactive element sulfur, and one bacteriophage reacted with the radioactive phosphorus. Then what happened? We can biological term, we can tell the leveled. You know, sulfur is a element which is present, sulfur, which is where it is present, you know, sulfur is present in the protein. Because you don't forget, the virus is a particle or organism which is consist of two components. One is nucleic acid, another is protein. Nucleoproteinol, that's why you are telling. Because it has only one, that is two are called the nucleic acid, and rest of the part made up the proteins. That's why when they taken the sulfur, because the proteins are made up of the sulfur, they taken the phosphorus because the DNA is made up of the phosphorus. Don't forget. They taken the sulfur radioactive because the protein is made up of the sulfur. They taken the DNA because they taken the phosphorus because DNA is made up of the phosphorus to make it radioactive. Now what happened? These two are become treated with the radioactive. Now they leveled with the radioactive. Simple term, biological term, we can tell leveled with the radioactive. Then the story what it proceed to, these two bacteriophage allow to infect the bacteria, because the bacteriophage they can infect the bacteria. You see this black color of this DNA is entered into the bacteria. That means infection. First phase of this experiment is infection. What is the meaning of infection? Any microorganism, when it will insert its genetic material to the other organism, that is called infection. In simple term, I would like to explain you. Suppose a person is infected by the HIV virus. What happened? The HIV virus totally it will not enter in our body. Only the genetic material of HIV virus become entered in our body. Like that, the when it is infected, the what bacteriophage is infected to the E. coli bacteria, it insert the genetic material here. And here also see its genetic material I marked as red, here red, here it is genetic material I marked, DNA marked as the black, here also black. That means this genetic material, what is present here DNA, it entered into the bacteria. Here the genetic material is there, it is entered to the bacteria. Now one question. Here I make the red here, but it is the black, because here what sulfur, when I will leveled, when I kept in the sulfur radioactive solution, the bacteria DNA is not affected. because DNA consists of phosphorus. But protein part is become this, all the parts consist of protein parts, which is the red one, they are become radioactive. Because when you will keep in the radioactive sulfur, only the parts will radioactive which is made up of the sulfur. That means protein become radioactive here. Here we kept in a solution of the phosphorus radioactive, because the part which is consist of phosphorus, they only will be get affected, because the radioactive phosphorus will affect to the DNA, because DNA made up of the phosphorus. Now you can try to understand what happened next when this radioactive element affected to the protein part, then it is entered into the bacteria, then the bacteria become getting infected by the bacteriophage. Then what happened, the next page, first infection is over. Infection means enter genetic material inside the bacteria. Second is blending. What is the blending? Simple electric mixing. What is the electric mixing? Means just to we will grind it by a help of a machine, that is electric mixing. What you mixed at that time when they will entered, because you should not forget, the all part of the microorganism which is infect will not go inside the host cell, only the genetic material will entered. Here this DNA entered, here this DNA entered, and after that when it is infected, we are become blending, means electric mixing. After mixing, what will happen? We are followed by a centrifugation. What is the meaning of centrifugation? Centrifugation means separation of the substances on basis of densities. Suppose here I will make a small diagram. You see here, if the solid particles will be here and the liquid part will be here, the liquid part become above and the solid part become down, because solid part become heavier, means more density. You see, that means which is called supernatant. Supernatant water, a liquid layer above the solid. Solid will remain down and liquid will come out. And in this, why we are doing the centrifugation? Centrifugation we are doing to separate the viral particles and the bacterial cells, because virus is affected to the bacteria. That's why we want to separate. When we separate, you saw this DNA come to the bacterial cell, which you after centrifugation we separated. What you got, the bacteria that are affected by radioactive. But after that, those who are become the in the sulfur radioactive level, they are not affected. What does it mean? Does it mean? Because when this bacteriophage, which is sulfur level, they are not at all affected to the DNA, because sulfur is made up of protein, and they are only the protein part of the bacteriophage is affected, which is not transmitted to the bacteria. Here what? Because radioactive phosphorus is affected by the radioactive phosphorus, the phosphorus made up of the DNA. DNA is made up of the phosphorus. That's why the DNA become radioactive and it will transmit to the cell. That's why the bacterial cell, after the centrifugation, we got two types of bacterial cell. One is which is treated by the radioactive sulfur, they are become the not radioactive, because they are not this DNA is not radioactive. That's why the cell is not radioactive. Here the DNA become radioactive because we are treated with the phosphorus, and phosphorus is made up of DNA is made up of the phosphorus. That's why the cell which is come after the centrifugation become radioactive. What you understand by this? We understand that actually Hershey and Chase want to prove that that what is the DNA material? It is a protein or DNA. They want to experiment that. Here, when they become treated with sulfur, the protein part become radioactive, which is not infected. When the infection is takes place, it is not bacteriophage is not insert the protein part to the bacterial cell. That's why they are not infected. That's why that is not a genetic material. But here what, when the DNA become get infected by radioactive element, first because phosphorus, because DNA is made up of the phosphorus, phosphorus become infected the DNA, and the DNA has found to by the E. coli. That's why it is proved that DNA is a genetic material, not protein. It has proved. I concluded my video by saying 2-3 words by which we can clear the concept. Here, the Hershey and Chase want to prove that whether it is a DNA or RNA is a genetic material. No, sorry, it is a DNA or protein is a genetic material. When they make the protein a radioactive, it is not seen in the bacterial cell after the centrifugation. But when the radioactive DNA, the radioactive bacterial cells become found. That's why it is found that when an organism is infected, it insert is a genetic material, and that genetic material seen in the bacterial cell E. coli. It proves that DNA is a genetic material. I hope you clearly about the concept, and thank you for watching my video.